Okay, so let's take what we have learned about DNA and we're going to apply that to biotechnology. So biotechnological techniques that are vital for molecular biology. We're going to be talking about PCR. And then later on in some additional videos, we'll talk about gel electrophoresis and bacterial transformation. PCR stands for polymerase chain reaction. Think back to what you learned about DNA replication and what you needed. So for DNA replication, you obviously needed DNA. You needed a way to break those bonds, which is helicase. Okay, so that was an enzyme. You needed you needed primers. And you needed DNA polymerase. You also needed ligase. Okay. That was for DNA replication. For PCR, polymerase chain reaction, we actually don't need ligase because we don't have Okazaki fragments to bind together. We don't need helicase because instead of using an enzyme to break apart those bonds, those hydrogen bonds, we're actually going to use heat. So let's look at this. And then the primers are a little different. They're not RNA primers. They are DNA primers. And the DNA polymerase is called Taq polymerase, T-A-Q. Now, Taq polymerase is extracted from a bacteria that is found in the hot springs. Right? So these extremophile bacteria, they actually took out this enzyme so that it can withstand heat because we are dealing with heat on the 3 different steps. So for PCR, there are 3 main steps. Okay, so the first step is called denaturation. And that's literally just taking your DNA. Let's say this is your small fragment of DNA. And increasing the temperature. So we're just going to say this is 96 degrees Celsius. You're increasing the temperature to break those hydrogen bonds. Okay. Second step, annealing. During annealing, you're actually going to lower the temperature. Say to 50 degrees Celsius. It's not very cold, but it's definitely a lot colder than 96 degrees Celsius. So you're dropping down that temperature to allow primers, and let's change the color on this. Okay, so you're allowing primers to bind to your DNA. So we have our DNA, and we have our primers. The last step DNA synthesis. All right, so DNA synthesis, you are synthesizing DNA, you're amplifying your DNA, which means you're making lots and lots of copies. So for right now, right, so you are you're actually increasing the temperature in your third step, not super hot, but definitely warmer than 55 degrees Celsius. So about 70, 72 degrees. Okay, Celsius. And so what's happening here, we've got our primers. And we have our DNA polymerase binding. And then the other thing that you do need with PCR that you would normally get from your cell. So this is all happening within a test tube. So inside your test tube, you have buffer. You have your DNA. Okay. You have your primers, lots of primers. And these primers are specific to the fragment of DNA that you are trying to amplify or create lots of copies of. You have your DNA polymerase. And then you have your nucleotides. So remember from the DNA structure, right? So you need nucleotides. And that's just your sugar, your phosphate backbone, and your base pairs. Now, because this has taken place within a test tube and not your body, you need to add those nucleotides to build along your strand. Okay. So after one cycle, you are going to get 2 pieces of DNA. You're going to repeat this cycle like 34 to 35 times, depending on your length of fragment. And that's going to give you 1 billion copies of DNA, which is super important for when you need this fragment, let's say for gel electrophoresis or medical testing. So it has lots of different applications.